Here we are. Okay, so now we'd like to invite David Lloyd back onto the stage. This is a joint presentation between him and Radu Grumeza, as unfortunately Radu is unable to be with us today. Thank you, David.
Thanks, Catherine. I think it's fair to say that this is really Radu's presentation. I can't take a huge amount of credit for it. Yes, he's not able to join us today, but so I'm presenting his slides. So if it seems like I'm reading off slides occasionally, there may be a hint of truth in that. Okay, so peas. It's fair to say that there's been a large increase in the acreage of peas in recent years. This is partially due to vast expansion in the vegetarian and vegan market, but also in the expanding so-called flexitarian market, where people are replacing a lot of meat in their diets with vegetarian type foods, but not necessarily going fully vegetarian. The use of high-value protein and starch fractions have contributed to this. So there's pea protein-based burgers and meat replacement products, beverages. We're beginning to see a lot more non-dairy type milks. So pea milk is being seen on the shelves. I have tried it. I'm not quite ready to recommend it yet, but yes, it's certainly drinkable. So there's pea flour-based pastas, extruded snacks, and also there's quite a lot of pea protein being used as fillers in sort of traditional meat products. So sausages with pea protein in them, and direct uses as foods. So cracked peas. Snacks, we can see a lot of snacks that sort of uses the same sort of market as potato crisp, potato chip type products. Mushy peas, beloved of chip shops in the UK, and canned dry peas. There's also a vastly increased usage for animal feed. As has been said in previous talks, we are very dependent on the importation of protein from North and South America, about 70% reliant on that at the moment. And pea is being used increasingly to replace some of the soybean imports, particularly in the northern parts of Europe, where homegrown soybean is not really an option. Okay. So pea genetic diversity. So within eucleg, we looked at 260 different varieties. These are extremely diverse, and we can see on the right hand side there's a dendrogram that's based on the initial genetic analysis on the right. I think it's fair to say that the goals within eucleg are broadly similar to those with soybean. So using GWAS and genomic selection to attempt to speed up the rate of genetic increase within the pea product, the pea germplasm. Majority of the cultivars used within the eucleg projects have been from core collections from the John Innes Centre in Norwich in the UK and Nordgene. Majority are dry pea cultivars of different origins from Europe, across the whole of Europe, America and New Zealand. So as you can see there's roughly about 120 dry pea types and various other types going from marrowfats, garden peas, land races and wild peas, and various others. We're at an earlier stage of data analysis than the group working on soybean are at the moment. We had a bit of a delayed start while multiplying sufficient material up for field trials. And it's fair to say that the COVID pandemic has delayed some of the data sets, particularly IBER's year 2 data sets. So we're only at the beginning of doing some of the genetic analysis of the traits that we've been measuring. So breeding objectives. I covered this in the talk earlier on today. We're breeding mostly for grain yield and yield components. So agronomic traits, plant morphology and phenology, so looking at traits that so things like harvest index, improving lodging resistance by having better standing abilities within the pea crop. Also looking at environmental and rhizobial interaction adaptations. We're breeding for protein content and composition, so looking at trying to improve the amino acid profile of peas. A lot of it, some of the branch chain amino acids are kind of lacking in some of the pea varieties, but we're finding some very promising germplasm within the eucleg collection that have increased protein levels and better amino acid profiles. Also looking at biotic factors, so disease tolerance within peas. The main diseases of interest are so-called root rot complexes, so that includes Aphanomyces, fusariums as well. Looking at Ascochyta, blight that affects leaves, Sclerotinia, botrytis, and Septoria, which form a canopied root complex. So quite often it's difficult to tell the difference between the different pathogens involved in that. Powdery mildew and downy mildew and rust, these are diseases which come in later on in the season. Downy mildew can be a real problem in terms of delaying maturity of the pea crop, and it has a major impact on harvestable yields. And also looking a bit of viruses, so things like let's see if I can remember the acronyms for these. So pea ination mosaic virus, pea seed-borne mosaic virus, bean yellow mosaic virus, which despite the name also affects peas, bean leaf roll virus, and the more recently identified pea necrotic yellow dwarf virus. So finding robust forms of resistance to these pathogens is a major sort of aspect of pea breeding efforts at the moment. Looking at abiotic stress, so drought and water deficit stress. So lack of water will obviously impact pea yields considerably. Waterlogging, which a lot of people don't really concentrate too much on. Peas are very sensitive to waterlogging. Certainly in the UK, the major area for cultivation is on the east coast, which gets far less precipitation than we get on the west coast. Some of this is down to the increase in disease pressure that you get in wet conditions, but peas really don't enjoy having waterlogged conditions. They suffer very badly from hypoxia caused by flooding or increased water content in soils. They're very prone to temperature stress, particularly high temperatures. Temperature above 25 degrees can lead to seed abortion, which obviously reduces the yield of the crop. And low temperature is not so much of an issue, except where you get frosts. So certainly in the more northern latitudes, certainly going to the sort of Midlands of the UK and above, we have major problems with yield losses through frost damage. They're subject to nitrogen stress, so I think it's fair to say that the nitrogen availability can have a major impact on rhizobial activity, and you get reduced yields through that. It's also linked with drought and water deficit. So the pea genome, it's a very large genome. Peas are self-pollinated, so we're obviously talking about pure line cultivars. The genome's 4 gigabases long, which is very large if you compare it to Arabidopsis, which is a genome of about 100 megabases, slightly bigger than soybean, which is about 1 gigabase, but I won't complain too much because faba bean has a genome of about 13 gigabases long, so absolutely massive. Sequence assembly that we have at the moment represents 88% of the genome. This is work that's predominantly been carried out at INRAE in Dijon, and contains 44,756 annotated genes. There's a lot of repetitive elements within this, including the wonderfully named retrotransposons Gypsy Ogre and Gypsy. I think that's a picture of the Gypsy Ogre over on the right-hand side there. The genome we have comprises 2 species, so Pisum fulvum, Pisum sativum. These actually can interbreed quite readily, so it's debatable as to how true it is that these are separate species, but also several subspecies, so Elatius and Abyssinicum. Genomic resources we use. There's a lot of marker technology that's available to us, SSRs and RBTP markers. So we use a fair bit of the markers that are available for various major genes, including things like trypsin inhibitors, flowering dates and lodging, resistance to diseases, seed composition genes. So there's vicillins and convicillins, which are major seed proteins which have an anti-nutritional effect, and we're doing quite a bit of work on that at the moment, trying to rebalance the balance between vicillins and convicillins and legumins, which are the more desirable storage proteins. So marker-assisted selection is being used quite commonly to integrate QTLs and genes of these types into the commercial varieties. It's inefficient, as I said earlier, for improving quantitative traits. So traits such as yield, which are polygenic, they have a lot of genes contributing to them. A lot of minor QTLs are very difficult to select using marker-assisted selection. So, and this is obviously compounded by previously low density marker availability. But recently developed SNP chips, so arrays have redressed this to some extent. So we've got now available to us 2 SNP arrays, so the 13.2 K SNP array that we used within eucleg, and a more recent 90K SNP array, which is proving to be very useful for increasing the density of marker coverage of the genome. And GWAS is really becoming the go-to method for using to improve the genetic sort of approach to breeding with MPs. So genomic selection was proposed in 2001 by Mewison et al. It's taken a while to get up and running because the density of coverage of markers that's needed for genomic selection is very high, and it's been very expensive to get that sort of coverage. It's still very expensive to genotype to sufficient coverage with currently available techniques, but we're nearly there. Prices are dropping constantly, and coverage is increasing at the same time. It can basically be looked at as a form of marker-assisted selection with extended scoping advantages. Genome-wide markers are used to capture genetic variations within the population and assign genome estimated breeding values. We use mixed modeling to do this, to assign the breeding values, and one of the technique which has proved most useful for us at the moment are BLOPs. So this is best linear unbiased prediction, which you'll be hearing about later on in the talks. So it is uses a training population to feed in data for traits and the genomic genotyping into the modeling, and we use that to predict the breeding values of plants. I'll show you some examples of the genomic predictions later on. Speed breeding is being used more often at the moment for peas than it has been recently. So Watson et al. In 2018 really sort of advanced this. It says here that we can get up to 6 generations per year. Well, that's true for very rapid cycling varieties, really for material which would normally cycle very quickly anyway, but not really so much for agriculturally useful pea varieties, which tend to be later. 4 generations is possible, but as I said earlier, this really puts a strain on glasshouses. Some downtime within glasshouse use is very important for keeping pathogen loads down. So genomic selection with segregating populations leads to yes, needs to be combined with classical pedigree selection or bulk selection techniques and multi-environmental trials, especially for qualitative traits. We use functional omics studies. We have now available gene expression atlases. RNA-seq is used for plant pathogen interactions, and gene expression studies are being used for FOMA, powdery mildew, and fusarium species. So the future of pea breeding, genomic assisted breeding is going to be used a lot more. Microassisted selection and integration of GAB with speed breeding, yes, particularly for genes that have major effects on traits. Genomic selection combined with high throughput SNP assays and high throughput phenotyping is going to be increasingly used. Microbiome interaction is something which needs to be addressed at the moment, and genomic editing. Genome editing is at the moment is a bit prescribed within the EU. We've had an announcement with in the UK recently that with Brexit, we're going to be it's going to be available certainly within England, not so much within Wales and Scotland, but it is a technology which will be advancing in the future, certainly within China and North America as well, especially. Phenomics, so bridging the gap between genomics and phenomics is quite an important thing. So high throughput phenotyping. This is not necessarily just the phenomics approach, which uses the sort of train track approach where plants are shunted through through phenotyping systems. We can use drones and distance visualization of traits. New resistant sources are being looked for within land races and wild material. A lot of the resistance to diseases has been selected out through the domestication of the species, and it sort of underlines the importance of maintaining biodiversity. Functional genomics and discovery of new candidate loci, very important. And yeah. So this slide shows some of the initial results of genomic prediction through genomic selection that we've been doing within eucleg. On the left-hand side, you can see a cloud of points there. This is basically predictions of crosses between different lines used within eucleg. The red dots on the right-hand side, that's crosses between one particular variety or accession with all other varieties there. As you go along the x-axis, this is yield, and we've got variance on the y-axis. So as can be seen, there's predictions of crosses between particular accessions that will predict very high yields and less so on the left-hand side, although some maintenance of variance is important in doing selections using this method. On the right-hand side, it's the same data set, and the black dots show another the contributions of another accession, which you may not want to take forward into breeding a new variety. So sort of underlines the importance of germplasm selection within this. And yeah, just like to thank you for listening. Any questions, I'd be happy to answer.
Thank you very much, David, for presenting on behalf. Oh.
Oh no, you're not.
Can you hear me? No. All right. Thank you very much, David, for that
my
presentation.
speakers.
We haven't got any questions for you as of yet. I don't know if there are any questions arising.